Three, two, one. This is Into the Absurd, episode 22 with Dr. Nkrumah Grant. Thank you for coming on.
Thank you for inviting me on your show today. So as Greg just stated, my name is Dr. Nkrumah Grant, and I am a postdoctoral fellow at the University of Idaho. I completed my doctoral studies at Michigan State University, May 2020, where I obtained a dual PhD in microbiology and molecular genetics, as well as in ecology, evolutionary biology, and behavior. Some of my other interests include working on different committees. So I'm currently on the program planning committee for the American Society for Microbiology. And I've also been involved in several instances in different venues related to diversity, equity, and inclusion.
So what all kind of research do you do?
So my research interests writ large is in evolutionary biology. And basically what I do is I use bacteria to study evolution in action. So that was some of my PhD work was based solely upon doing that. Bacteria are ideal organisms to study evolution in action because they're very, very small, which means that they require very little lab space to grow them. They have very, very pure nutritional requirements, so we know how to grow them relatively quickly. They can be frozen and revived. They have very rapid generation time. So where a human has a generation time is on average about 15 years or so, bacteria can double about every 6.6 generations per day, depending on media, or every 20 minutes, depending on the type of media that they're using. So I use bacteria to study evolution in action, and I really am interested in metabolism, the evolution of metabolism, how selection on different reactions can allow an organism to thrive in an environment, and how those traits are inherited over time.
So when you're doing this research, do you see the bacteria forming new species?
Oh, no. So evolution doesn't work in that way, in the sense that we can visualize these things over a long time scale. So you have to remember the Earth is, 4.6 billion years old. And if we were to look at the evolution of, say, like human primates or humans ourselves, we would have evolved in the last second of the last hour, right? So very, very short time spans. Speciation from a microbiological perspective, when we think about bacteria, is actually very, very hard to describe that, just because of the different concepts in biology that we have. However, we do see that adaptation occurs, and we know that because we can take more evolved strains from our evolution experiments. So during my graduate studies, I worked in the lab of Dr. Richard Lenski, for example. We could take those bacteria from now. We can, grow them in environments in competition with bacteria or bacteria from back in the day, 30 years ago, for example, and we can see that they get better at growing in these environments, for example. We do have some interesting things that also evolved. I have seen that as well. So for instance, one population in this experiment, and I can go in and describe how that experiment works as well later on. So one of these populations in this long-term evolution experiment I briefly just said something about, evolved this novel metabolic ability, that is the ability to utilize a carbon source that none of the other populations could use, including the ancestor and E. coli in general.
Wow. So you're kind of observing them being more efficient with their metabolism, essentially, than previous generations.
Yes, in this case, for sure. Definitely. Mm-hmm.
Wow. So how does this experiment work?
All right, great. So during my graduate studies, and mind you, I would also talk about and speak to what I'm doing here at the University of Idaho. I'm working in a completely different system. But during my graduate studies, I worked with E. coli, right? So E. coli is a commensal gram-negative bacteria just found ubiquitously everywhere. It typically doesn't cause disease, though there are some strains that can cause disease, for example, and cause diarrhea and so forth. But I used a non-pathogenic strain of E. coli, and particularly my advisor in 1988, he took a population of E. coli, a single cell, an isolate, right? And he derived 12 populations from it. So he grew them up, then he had 12 replicate populations. 6 of those populations, if we plated on media that allows us to differentiate between their metabolism, it forms red colonies, and the other 6 forms white colonies, right? So you can imagine now you have these two separate parallel parallel populations with different colors that we can use to now, say, for instance, in the future, compare red to white, given the earlier strains and descendants. So every day, someone goes into the lab and we take these from these 12 populations from the flasks that have grown using up all of the glucose, and it's a minimal glucose environment, glucose being the carbon source. We take 1% of that culture, we transfer it to new media, it uses all of that glucose, and in that time period, it undergoes about 6.6 generations per day. Then we transfer it again and transfer it again, transfer it again. Every 500 generations or so, which is roughly about 75 days, we take samples from those populations and we freeze them and we store them, which creates a system where we can go back, and instead of kind of relying on fossil records and these type of things, which have historically been used to make evolutionary inferences, we can directly observe the ancestors and descendants of those ancestors in real time. We can look at them using things like microscopy, which is some things that I have done. It lends very well for the generation of novel techniques. So, for example, genome sequencing became readily used, or at least cheap in some instances, in about the year 2000 with the Human Genome Project, right? So now we are able to subject these populations to large scale, grand scale DNA-based experiments and make inferences based upon that. Also, we can do things like transcriptomics, where we can look at gene expression in these organisms now for relatively cheap, and so forth, right? So it lends well to kind of like creating a space, or space holder for future analyses and technologies as they become developed.
Yeah, that's really cool. I mean, observing evolution in real time, especially with, all the people that don't really believe in evolution, right? And well, hey, we got it right here.
Exactly.
See it right before your eyes, And I guess, most of the time people are kind of not really, they don't really understand the full picture, like how it takes millions of years to see evolution of a multicellular organism take place, right? And how, say, I was talking with this philosophy professor, and he did some research on how when we describe things, it's there's a lot of things that don't really have a spot where they can be easily defined, like a heap of sand. Like if you kept removing sand grains from a heap of sand, at what point will it not be a heap of sand anymore, right? And this can kind of be applied to human evolution in the sense where, well, there can't really be a place where there was a first human. There was kind of maybe an in-between area, but there wasn't really ever one single point where, oh, this was the first human, right?
Right. That said, we can use the fossil record and evidence with modern DNA technologies to say that we at least derived from, this last common ancestor, for example, right? Yeah, exactly. So yeah, there's definitely gray areas, right? That's, these incomplete fossil records, as I'm stating, right? And we found, evidence of, even the common ancestors that we find evidence that, there exists sometimes earlier forms that could be representative of our lineage, in general. But yeah, that's what makes using bacteria an ideal system to study evolution, right? Because again, we can go back into the freezer at any given time and we can say, hey, this is exactly where this strain come from. Look how different they are. Look at what it can do versus its parent, for example. So,
yeah, things. It's things change, things adapt.
Exactly.
So what do you want to do with this research in the future, your career?
That's definitely complicated. I guess, long long, big picture goals is to become a professor at a university. So I'm definitely interested in academia, right? Teaching at the university, being able to continue research and to train the next generation of scientists, right? So that's definitely what I would like to do. I have, began to start thinking about, like different, career focuses in industry or, something thereof, you always need to have a backup plan, right? So, but yeah, big picture goals is to, become a professor at a university. Ideally, so I guess I could start in, talking about what got me into science in the first place, right? So I'm very, very much so interested in life outside of, our planet, right? So as far as we know, Earth is the only place that has life, right? However, we start thinking about, different. We're starting to find planets that has the conditions that mirror Earth, for example. And for example, we have Perseverance on Mars, looking for extant life, right? Exactly, right. We believe that Mars once was habitable and could have been inhabited by microorganisms at least. So I'm very much so interested in using experimental evolution and developing evolutionary theory, not only to, and coupling that with actually synthetic biology, to actually not look for life that may be existing elsewhere in the universe, but actually create or generate life that could be seeded at different places in the universe, right? And we have organisms, in several extreme environments, highly radiation resistant bacteria like Gonococcus radiodurans, for example, or halophiles, organisms that live at high salt concentrations, organisms thermophiles that live at high temperature extremes, for example, right? So we have organisms that live in these strange places here on Earth. And because we have technologies that allow us to cut and paste genes from organisms, and organisms are pretty plastic in the sense that they can, take up foreign DNA and keep it for the most part. Some of them, of course, um, we can actually begin to cut like Legos and build organisms that could live in Mars and be, at high CO2 concentrations, for example, right? So definitely want to use it for more of a, applied side, the evolutionary roots and synthetic biology roots that I'm generating now.
And with this, I know some organisms over time can essentially build a atmosphere that's suitable for other organisms, like with the algae boom of whenever that was. There was a huge algae boom that allowed for cellular respiration to occur in life forms, right?
Exactly, right. So, I mean, think about, we breathe oxygen, which is a waste product of plants, and cyanobacteria, right? So if it wasn't for the cyanobacteria, these are small little bacteria that inhabit, marine ecosystems, for example, there wouldn't have been enough oxygen to support, oxygenic oxygen respiration, for example. Organisms that respire using oxygen, right? So organisms are always, producing metabolites and excreting them into the environment that then serves as, different substrates, or at least has the potential to serve as different substrates for other organisms to take advantage of. And even in, some of these experimental evolution studies, while we are feeding the Escherichia coli, for example, glucose, their waste products include something like acetate, right? And then we see these environments, these microenvironments in these flasks where you have some of the bacteria specializing on utilizing the glucose, and other bacteria specializing on using the acetate, right? So you're always going to have this niche construction, going on, right? That opens up opportunity for other organisms to exist and evolve.
Mm-hmm. Yeah, I mean, everything kind of plays its role.
Exactly. Right? Mm-hmm.
Do you ever delve into environmental philosophy at all?
Not necessarily. I mean, I do think about it. So some of the work that I'm doing right now. So currently I work with this species called Methalobacterium extorquens. It's a plant-associated microbe. It lives on the surface of plant leaves. It's also found in sewage and in soils, etc. And it's unique in its ability to use C1 compounds. So C1 compounds are, so glucose, for example, has 6 carbons. A C1 compound will only have 1. So something like methanol, for example. It can use methanol for growth. And in doing so, it also takes that methanol and assimilates it and make larger compounds, right? So, we kind of use this organism as a chassis for feeding them methanol, these basic compounds, and then building bigger molecules like plastics or butanol that can be used, to power cars or whatever, or to make other products, right? So that's kind of like what we do with that. And ideally, what we would like to do is be able to feed this microorganism detritus, so plant cell wall material, and then build some of these things, right? So to kind of, have, instead of, the reliance on fossil fuels, but using, bacteria and plant waste, et cetera, to make something that we could potentially use to, bring down carbon dioxide and other gaseous compounds in the atmosphere.
So you're essentially looking into how you could use this organism to clean up the atmosphere a little bit.
In some way, yes. It wraps back around to that, right? Because we're using, we're using an organism and product that can be regenerated. Yeah.
Mm-hmm. So with, say, building an atmosphere on Mars, do you have any ideas of how we would do that in the future?
So that's very, very complicated, right? So I'm not a physicist or a planetary scientist, but I will say that when I first started graduate school, that I took a chemical engineering course, and that was one of the things that I for one of my projects, I thought about how could we build an atmosphere. Obviously, building an atmosphere or having an atmosphere requires, some type of gravity that keeps the atmosphere, right? So, or, so for instance, here on Earth, we have this, the rotation of the Earth's core that's generating these electromagnetic waves that is creating our atmosphere. And we have gravity that's strong enough that's, maintaining the atmosphere. I think any planetary scientists don't jump on me if that's the wrong description, but I'm pretty sure that's how it works. Mars, it is believed that it once had an atmosphere, but the core, it's not rotating as much. I believe it's a dead core at this moment. So the atmosphere over time was stripped by solar rays from the sun. All right. So for it to make an atmosphere, we have to first and foremost have a way to contain the atmosphere. Otherwise, you're going to make it and it's going to be lost in space, and that's not productive at all. So that would be one step. Secondly, you have to decide whether or not you're going to have create an atmosphere that uses, technology, right? And that's expensive, right? If I remember, a couple years back, to send 1 pound of anything to space, I believe it costs about $10,000, 1 pound. So that means for a person as big as I am, it costs at least $220,000, right, to send me to space. That's a lot of money. And we have these machines, if you want to build anything there that you would want to send, et cetera, it's just not feasible in that way. It's going to be too expensive, and of course that expense is linked to hydrogen used to power or propel spacecraft, jet fuel. So, that'd be expensive route. Another way, if you, independent of trying to send things to space, Mars has very, very rich metals, it's full of metals in its soil. You could send something, right, that's able to, collect dirt and make it into the things that you need, right? So like, send something with 3D printing capacity, for example, that could print more copies of itself, and then print, the machine that you need to convert, CO2 or to generate an atmosphere that would keep some of these gases in. But that would also be, a technological feat that'd be difficult. You can also build little spheres. So instead of, relying on, gravity from the planet, et cetera, you could build domes, for example, and then make atmospheres that are contained at least within the dome. And that way, that seems a little bit more reasonable to me. And you can do that. And then that way you can breathe within those domes, just fine. So actually on the new rover that was just sent, there was a machine that was, it's not necessarily there to, generate a new atmosphere, but it's there at least to see whether or not it can take Martian carbon dioxide and convert it to oxygen for human missions, right? So I think that's something cool there. Ideally, it would be nice if you can use bacteria or something, right? And if you can use bacteria, take the CO2, then convert it to oxygen, you have something that is, sustainable in a way. But they need nutrients and they need, energy and stuff too. So I mean, it's complicated no matter where you go on how to do it. But yeah, those are, I think, the basic requirements that one would need to, I guess, terraform a planet.
But theoretically, if there was a place on Mars where there was nutrients that bacteria could survive, it could be done, right?
Exactly. Yeah. At least in small at a small scale, right? I can't say that you make a atmosphere over the whole planet based upon a pocket of bacteria living in a crater, for example, right? So the benefit of why oxygen was able to dominate our atmospheres again, because we're able to retain that oxygen, and because the oceans is the ocean, right? Constitutes about 78% of the world, right? So you have this high dispersal of organisms creating oxygen, nutrients to its advantage all over the place, right? Always being input into the system, right? So, yeah.
But I don't think they really found a whole lot of nutrients on Mars.
Yeah, exactly. So that would be a difficult feat, right? So how would organisms? I mean, the sun, right? So the sun is a nutrient source, right? So you think about, but what's important is like some kind of liquid water, right?
Yeah.
So, yeah.
Yeah, and I guess the problem on Mars is, well, without an ozone layer, there's just so much UV radiation, and it's also extremely cold. So it's kind of it's like an icy hot scenario.
Exactly.
So have you delved at all into CRISPR technology?
So I have not actually. Something that I want to do. Obviously I know quite a bit about it. Actually, when I first started graduate school, that was like, I mean, it's still a big buzz, right? I mean, the tech has opened up opportunities to a lot of stuff. So like when I was saying, this cut and paste type idea, right? With synthesizing organisms, right? A lot of it is done now using CRISPR-Cas9 based systems, right? So yeah, it's definitely a big thing. So I haven't used it yet. It's something I do ideally want to use, in the future. But I haven't used it.
Could you explain CRISPR a little bit?
Right. Yeah. So CRISPR actually, while it has the media's attention or this buzz about being able to use it to take genes and make edits within the genome, for example, it's actually a bacterial system. It's actually a system that bacteria use to prevent invasion by bacteriophages. And these are viruses that infect bacteria. And how it works is when a virus or bacteriophage infects a bacteria, it injects its genome into it, right? And the bacteria says, okay, well, it says, I don't want to anthropomorphize the bacteria, but the bacteria responds in a way that says, okay, here's a foreign invader. Let us pick some of this copy of this invader, right? And now we're going to integrate that piece of DNA, these small pieces of DNA, into our genome. Once we integrate that into our genome, right, we now can recognize this this virus that is invading the cell based upon this unique genetic signature. And we're going to take our proteins, right, and we're going to cut that virus there, and it's going to kill the virus, right? So that's basically how CRISPR works from a bacterial perspective in the way that it evolved. Now, this was found by Jennifer Dauda. Please, I think, Jennifer Dauda, I believe. I'm sorry, definitely 2014 is a long time. She discovered this system, these repeating units within the genome of bacteria, And she said she didn't know what was going on. What is this, right? And then she found that that's how CRISPR-Cas9 was discovered. And then she found that we can take sequences in the same way of a gene, any type of gene, across organisms, right? And now we can target that gene, and we can either we can cut that gene out if it's something that's bad. We can actually replace a mutated position in that gene that's causing disease, for example, and revert it to the wild type non-disease allele or alternate form of the gene, for example, and change it back. And it's been successful in that way. So it's actually a bacterial defense system that we have co-opted or have exploited, right, to use to create for genetic manipulation.
Yeah. Yeah, and this could, I mean, at least I watched this Netflix documentary on it, or part of it, and it seems like it has the capabilities of essentially healing almost all human diseases.
It could, but so it can, right? However, there's a time point in which that would be more likely, right? So you have to think. So bacteria, for example, there's single cells. You can make a change in the bacteria, and because bacteria divide by binary fission, that means that they split and make two of themselves, right? Perfect identical copies of themselves with mutations and things that in small minor changes in expression, or you may have a mutation that occurs at some rate relative to that bacteria. But in a perfect world, they split and create a perfect copy of themselves. So you get the immediate inheritance of those changes by a daughter cell, right? And that's actually quite different in humans, right? So the only time that in other evolved eukaryotes, for example, the only time that we can multicellular eukaryotes, and this is because we have our germline, we have our somatic lines, right? So germline, our sperm, our egg cells, for example, eggs are inherited from in women, and they're there and present from birth through the end of life. Males, we regenerate, our, gametes, for example, we every so often, millions upon millions upon millions upon millions, every day, we're constantly regenerating. Now, once, with sex, and once you have, the fusion of these gametes, right? Now you have genetic material that's copying. One cell becomes 2, 2, 4, 4, 8, et cetera, et cetera, et cetera. You have this exponential growth of these cells. And it becomes very, very difficult now to take a system and then target all of those cells, right? And make those changes, right? So you get this patchwork of changes that would occur. Ideally, if CRISPR is to work, in the best way, in humans, for example, to treat disease, you would want to identify the disease early on, right, during reproduction. So maybe do a screening of an egg and screening of screening of these two cells once they're together. Make those changes then, right, before the cell is allowed to develop into an embryo and go forth from that way, right? So yes, it does have the power, but there's a time in which you'd want to use or to use that technology to its full advantage.
Yeah, I guess kind of even if you cure the disease, right, it can still pop up in future generations.
Right, in a different cell if you start later, right? But again, if you started early, right, within the development process, right, it's like that individual will have that individual in itself won't have that mutation or suite of mutations causing that disease. And because that's going to be relatively stable over its lifespan, that individual's lifespan, when it mates with another individual, it won't at least pass over that disease allele, right? Now, the other individual could, if they have, a mutation or an allele or some disease, right? They still could, right? And again, we get into dominance and, recessive, for example. But yeah, it would be primarily gone from at least the individual in which some of this technology was applied to.
So ethically, there are kind of these two moral predicaments that are kind of involved with this technology. One, there's people that are, well, it's immoral to say mess with biology, right? It's immoral to mess with genes. And then two, there's, well, if we have the power to cure genetically caused cancer, then it's immoral not to cure it, right?
Mm-hmm.
So what do you think about that?
I mean, so I definitely fall on the right, the second, right? So on the latter side of the coin, right? I do feel like we have a more responsibility if we can, as conscious, bright, humans that we are, to fix things if that's the case. I mean, the medicine, right? So I have a headache, I'm going to take some Advil or Aleve or Tylenol or whatever I'm going to take to fix that. God forbid I get cancer, right? I'm going to go and I'm going to seek chemotherapy or I'm going to seek radiation, right? So that, I can cure that disease. There's so many, my ankles, my whatever, I'm going to go see a joint specialist, right? To fix those things, right? I definitely believe that if we can do preventative care, right? Why not? You understand?
Mm-hmm.
As opposed to fixing the problem once it's there. I think more of a bigger ethical concern is who has access, who would have access to some of these treatments, right? So you can imagine a scenario where someone goes in. There's a famous movie, like a very, very old movie called Gattaca. That was very, in the late 80s, early 90s, that came out. And it was about this exact thing. Imagine going, and saying, okay, I want a baby with, brown eyes, very dark skinned, tall, slender, 220 pounds, no bigger than that, all muscle, whatever, right? You can go and do that. Then you then begin to create, these super humans or super, you just get to really basically buy or purchase, the traits that you want, right? You want someone who is going to have an IQ of Einstein, right? What is that going to create? That'd be cool, right? But what is that going to require? It's going to, those with money, right? Those with weight, those with connections, whatever, however you want to say it, are going to be the ones that's going to have access to that, right? Mm-hmm. So then it's going to further stratify, right? The elites and rich, poor, whatever. And I think that is a bigger, concern than to say fixing or curing a disease is, because we do that all of the time, right? We do that all of the time. We've been doing that since medicine, right? Came about with the Medici family during the Renaissance. So,
yeah, I guess, with this technology, the rich are not only going to be rich, but they're also going to be not susceptible to any disease, super strong, and extremely intelligent. So
right, and yeah, the cards, that's stacking the deck, I would say.
Yeah, it's kind of, it's kind of the argument. Well, this technology would be good in the hands of Gandhi, but maybe not in the hands of Hitler.
Exactly, right?
Yeah,
exactly.
But that kind of goes with everything.
Yeah, that's true.
Like nuclear power, because we could make nuclear bombs, or we could make nuclear fusion reactors that power the whole Earth with just a drop of water, right?
Exactly.
So,
right, you're always going to have opportunists or people who capitalize upon some tech or some feature, right, with evil intent. You're always going to have that, unfortunately, right? But that's human nature.
So when I was watching that Netflix documentary, though, there were people using CRISPR like in their basement with like $50 worth of equipment.
Yeah, so I like to tell people, like, scientists, you see us and we may, present these very, very, big, complex ideas. And of course, it's, based on literature, and it's based upon previous observations and our ability to take and dissect that information and interpret it in new ways, or, design experiments to answer new questions, right? But science is, actually pretty simple, right? It's pretty smooth in a sense, right? And I say that, maybe I'm biased, right? Of course, I can say I've had things and have done things that are extremely hard. But it's pretty simple, right? If you read, you study, you have, the internet is here at your disposal. Papers are often open access to access some of these journal articles. For example, protocols are around, right? So you can do stuff, and I think you have these, I don't want to say citizen scientists, but garage-based scientists, right? Who are, doing things, right? I don't know if they're necessarily doing anything great in terms of, I don't know what their intentions are, right?
Mm-hmm.
Of course, it won't be published, but still, yeah, I mean, I think it's, it's at least accessible in a way that allows people to do things, simple, right? So, yeah, it's possible.
Yeah, this one dude was trying to make his dog glow in the dark.
Yeah, that's unethical. That's unethical. You don't know about what side effects are going to exist or, be caused from, doing some of these things, right? So, and that's another thing. For example, there are some 2 children who were secretively, they used CRISPR on these embryos, right? Very early on. And now these children they can't contract HIV, right? So they don't have the receptor for that, right? Oh, okay, they shouldn't have done that. We have, there are ethical concerns there. But one side effect of that in this case is that this same receptor, if I remember the story correctly, is also involved with intelligence, right? So it may be that these children, just by virtue of, having this mutation or having these changes done to, reduce, to decrease or reduce, get rid of susceptibility to HIV, for example, now they may have an advantage in IQ, right? So, and of course it was money from that family that gained them access to that type of technology, right? So again, that goes into the ethics I was talking about earlier, right?
So they're not susceptible to HIV and they're more intelligent?
They have the potential to be. The potential, right? We don't know, right? Of course, intelligence is, is a combination of things. There may be genetics. I don't believe in that argument too much, but also there is environment and including upbringing, schools, where you go, whatever, right? So yeah, they have the potential.
There's also multiple kinds of intelligence.
Exactly.
Right. There's a spatial intelligence, being able to get around, symbolic intelligence, being able to represent things in your mind, like mathematics. I guess like just street smarts in general.
Yeah, exactly. Yeah, there's multiple levels to intelligence. I definitely agree with you.
So is there any other ethical concerns that comes up in your research?
I can't say that. I mean, in my own personal research, that there are ethical concerns. Of course, there are environmental hazards working with some of the things that I work with, and stuff of that nature. But in terms of ethics, because I have access to different chemicals and different technologies, right? There's always that personal ethical responsibility to make sure that I am doing things, in a way that wouldn't cause harm to, the public or, myself, for example. I can't say in terms of any grand scheme, big picture ethical concerns, at least with what I do, right? That I've had to worry about that too much.
Cool. Well, I think that sums it all up. Is there anything else you'd like to say?
No, I think so. I would just like to thank you again for inviting me to come and talk with you today and just share, a little bit of what I know and a little bit of what I do. People can follow me at Nkrumah G on Twitter.com. And again, I'm at the University of Idaho. So yeah, that's about it.
Awesome. Yeah, thanks for coming on.
All right, thanks, Greg. Appreciate you.
If anyone has any questions, just email into.the.absurd.podcast@gmail.com. Thank you for listening.